Hi everyone, my name is Anu. I'm a final year undergrad at the University of Saskatchewan, and today I'm going to be talking about some interesting patterns that we found in gene expression in astronauts. Okay, so to give you a quick overview of what we're going to talk about, I'm going to quickly mention some of the issues astronauts face and the problem we set out to solve, and then talk about our methodology, go through our results and conclusions, and then I'll be glad to answer any questions soon. So most people here have a really good understanding of this already, but it's still worth briefly mentioning that spaceflight negatively affects pretty much every single system in astronauts, both at the organism level and also at the cell level. From muscles to pleural to cardiovascular to even the immune system, it's all very, very heavily affected, and most of the time it's negatively affected. And we know that these negative effects happen, again, at both the organism and cell levels, with spaceflight-induced mitochondrial dysfunction being a main cause of all of these negative issues at the cell level, specifically. But our lit review revealed to us that a large portion of current research in the field focuses on either individual genes or gene families. And there is research, but there's very little looking at gene expression as a whole, the mechanisms of gene expression, and co-expression patterns within the genome. So that's kind of what we decided to focus on for this study. So because spaceflight is so expensive and difficult, one of the main issues that we face in space medicine is the number of samples. The twin study is a famous example of this. It's one of the most detailed analysis ever done on the human genome, but it's only an N of 1, right? So the conclusions we can draw from that study are very limited. And so for that reason, we also attempted to develop a way to combine multiple gene lab studies for this research. So we started off accessing raw data from NASA Gene Lab from 5 studies that had both untreated control and untreated flight samples, and then internally normalized and reprocessed these samples with themselves. After that, we did basically a whole bunch of analysis on both the combined and the individual data sets to verify our normalization and data combination methodology. So this is, again, just a graphical overview of the methodology we use, as well as some info about the individual data sets. It's worth noting here that we used a mix of cell types, and we did that on purpose because we wanted to look at whole organism-wide responses, not just tissue-specific responses. So moving into our results, the first thing we looked at, of course, was to verify our normalization method. So we did PCA to, again, verify and make sure our normalization method was okay. So when we colored the data points based on data set, I think it's very clear to see that you have clustering based on the individual data sets, which is, again, what you'd expect to see. You're not going to have multiple data sets, multiple experiments clustered together. And so these bad effects are based primarily on mission profile. But post-normalization, we see a lot more uniform distribution of these points, which indicates, again, very good normalization. Now, when we take the same graph and we just color it based on flight and in-flight samples, what we see, again, is quite significant grouping of the assay type based on, assay type is the shape. So you see quite significant grouping based on shape, especially in PC2. But post-normalization, we see a more even distribution, which is, again, really good. We also see something really interesting in the grouping of flight samples together right in the middle of that graph there, which implies that the analysis that we do further on is really good at analyzing the flight and ground difference. So now that we've done this analysis and verified our normalization methodology, of course, the first thing we do is differential expression. So what we looked at after the differential expression is pathway analysis to see which genes and pathways change their expression, because once you combine data sets like this, you cannot then look at individual gene expressions, because that's just not valid. So here I've shown you the top 15 pathways, and 12 are very, very heavily involved in mitochondria and energy processing, again, functions which are already known to be very heavily affected in spaceflight. Especially EGFR1 and IL-6 were talked about quite a bit in both talks at this conference, so it's good to see that overlap. Now, before we continue on to the main meat of the study, I'd like to take a second to explain gene pair correlations here, because this is what the rest of the study focuses on. So if you plot the expression of gene A against gene B on an XY plane here, correlation between two genes basically tells you how likely you are to be able to predict the expression of gene A from gene B. So it tells you the relationship between those genes when they move. So positive correlation means that when gene A goes up, gene B goes up. When gene A goes down, gene B goes down, which means that they move in the same direction. Negative correlation, which is here and described in red, basically means that when gene A goes up, gene B goes down, so they move in opposite directions. So you'd expect to see positive correlation in something two pathways that perform a similar task, and negative correlation would be two antagonistic pathways. Zero correlation also just means that you cannot predict the expression of gene A from gene B. So what we then did is map all of these correlation profiles onto colors, right? And so the darker the color in general, the stronger the correlation, and the closer to white the color, the weaker the correlation. Also, the more blue the color, the more positive the correlation, and the more red the color, the more negative the correlation. And that scale is shown right there. Sorry, I think it was there. Yeah. Okay. So this is what a correlation plot looks like. Essentially, this is a list of genes plotted against itself. And at each XY intersection, we draw the dot that corresponds to the relationship of those genes. So this is only about 30 genes. And here you can see like a wide variety of correlation profiles. But what happens if you do this for the entire genome? And so that's what we did next. So this is the ground data set. And here you can see we did 10,000 genes. Interacting with 10,000 genes, which is over 100 million individual gene-gene interactions, which I think is really cool. But when you count replicates with the individual emissions, you count data sets, you count samples, there's over 10 billion data points represented in just this one image, which I think is really, really cool and really interesting to point out. Now, something to observe here is that pre-flight, gene-pair correlation is in general quite strong, which is what you'd expect, right? So absolute value gene-pair correlation being strong just means that the graph in general is going to be darker, either dark red or dark blue. And again, you'd expect to see that, because the human body is a very organized system and pathways work together to accomplish tasks. So you're going to have antagonistic pathways be very strongly negatively correlated and synergistic pathways being very strongly positively correlated. Now I'm going to move that over to the left and do the post-flight analysis. And so as soon as you bring in, excuse me, this is not post-flight, this is in-flight. And as soon as you bring in the in-flight samples, you see a huge change in gene-pair correlation pretty much all across the genome. So this implies that not only is gene expression significantly changing, but the genome is also starting to, well, we think it's starting to lose regulation in how the genes move with each other. And so the genome is starting to lose coordination as a whole, and possibly even activating conflicting pathways at the same time, potentially causing a lot of the cellular level issues and stress and everything that we see in astronauts. So something else that I want to point out before I move on is and another thing that we did in these plots is clustering of these 10,000 genes to identify if correlation loss is something that happens in any specific pathways. And the way we did that was a naive mathematical clustering of this correlation matrix. So we did not introduce any biological bias into this data set. And what we found is that these correlation clusters, for the most part, don't really overlap with any biological pathways, which is really interesting because that means that genes that aren't classically known to be identified or interacting with each other are somehow losing coordination in the exact same way to go down to that loss sample there. So this implies that there are spaceflight specific pathways in which correlation is lost, which I think is really, really cool. So speaking of pathways, we also replicated this in just neuronal genes. So we did a bunch of individual pathways, but I'm just showing you an example for neuronal genes. And so here there's only 300 genes, so it's 300 by 300. And what you see is a similar correlation loss. And it's worth noting here that we have a stronger absolute value correlation here, which again you'd expect to see because neuronal genes are going to be more closely related to each other than neuronal genes that, for example, a gene related to the gut, right? Yeah, so what we then did was network level analysis, both at the microRNA level, which is shown here, and the protein-protein interaction level, with the highest differentially correlated genes as well as the highest differentially expressed genes. So we created a cutoff based on false discovery rate and correlation value as well as the differential expression value. And so the genes that are the most differentially correlated, the most differentially expressed, and also have the lowest FDR are used to create this network. And what you see is a very, very dense network, which implies that it's possible to have changes in a few top controlling genes that are the most interacting that then trickle down to the rest of the genes in this network and then further on trickle down to the rest of the genome. We also did functional analysis. I apologize, that label should have been a bit bigger, but the main functions that we see altered in the microRNA level are mitochondrial dysfunction, inflammatory stress, stuff that's already known to be very, very heavily afflicted in spaceflight. So this is the protein-protein interaction slide, and again, you see the same story here. Very, very dense network of interactions, which reaffirms our conclusion so far within the protein expression proteome as well. So I know there's a lot of data and figures that I just showed you, so I condensed this into 3 take-home messages that I think encapsulate most of this research. So firstly, that gene pair correlation loss in a universal manner is possibly a central characteristic of spaceflight and astronauts, because we've done this in 5 studies so far, well, 6 studies so far, and we validated this in individual studies as well. We haven't seen anything like this in any samples for any diseases on Earth, and this kind of large-scale genomic alteration without necessarily changes in gene expression, because remember, you can have changes in correlation without changes in gene expression, is something that's completely unique to space as far as we know. Secondly, it's possible that spaceflight induces unique biological pathways that are only activated in spaceflight and that are different to the biological pathways that we know right now, because genes clustered together in correlation clustering, just through naive mathematical clustering, don't necessarily overlap with biological pathways. And lastly, that correlation breakdown tends to overlap with known degenerative markers and health losses in astronauts, and so this presents an exciting diagnostic monitoring and treatment opportunity for astronauts in space. So before I wrap up, I'm going to take a quick second to acknowledge all of my collaborators and supervisors, Lauren and Sylvain, who've been absolutely incredible with their insight, advice, and direction, as well as Dr. Lukong, Dr. Kanbegu, and Dr. Yang at the University of Saskatchewan, who were instrumental in the success of this project. Of course, the project wouldn't be possible without the open data that GeneLab provides us access to. So a huge shout out to the GeneLab and OSDR teams as well. We're in the process of publishing this paper right now, but the preprint is accessible through the QR code there. Yeah, we also ended up having some fun in designing a patch for the experiment because we're working with some space data, and I almost always have some stickers to hand out, so come talk to me afterwards and I'll give you the sticker. Yeah, with that, I'd like to say thank you for everyone for your attention, and to the symposium team for giving me the chance to talk about some of my work. I hope you found it interesting, and I'm looking forward to answering any questions. Thanks.
So the time only for one question, please.
That's okay.
Anyone?
Questions? Also, a quick question. Actually, kind of common. Because I mean, we have been thinking about all this correlation and everything, but then all the information we learn on Earth is like you trigger this or you disturb this gene, and which causes a change of other genes and everything. But we are in space, and especially after human or any biological systems adapt to an environment, they reach a new equilibrium. Say they basically lost all the correlation or the information we learned on Earth. So just wonder whether, I mean, in this kind of analysis, you'll be able to get some consolidated information out of it. I mean, in addition to
Yeah, for sure. So in the preprint, we actually go into more detail about this. But what we've identified is from the most differentially correlated and differentially expressed genes, if you keep increasing the cutoff that you use, the cutoff level, you end up with a list of like about 50 genes that we think are like the most detrimental, because all of those genes exist. Some of them are protected as you have like a lower cutoff, but as you increase the cutoff, most of the genes start to have detrimental effects when they're altered in the ways that we see. So of course you need to identify which of the genes you're looking at and which of the responses you're looking at are actually beneficial responses, right? And are the body like protecting itself and adapting to spaceflight? But from our analysis, as far as we can tell, at least for these data sets, as we increase the cutoff and once you reach a certain number of genes, all of the genes are negatively affecting your response. And so that analysis is in the paper, and so I can forward that to you as well, and we can connect afterwards.
Okay, thank you.